So much for the invitation to give this talk. So today I'm going to talk about a paper that we published just a couple of months ago, which aims to address the underlying physical mechanisms that give rise to DNA methylation patterns in healthy and in cancer cells. And what I'm going to do is I'm actually going to talk first for the first 25 minutes about some underlying theory and give you some historical perspective on some experimental measurements and some theory that we developed to understand those measurements. And then I'll proceed to explain how the fundamentals of how proteins translocate along DNA is related to the methylation patterns that are measured in these cells. Okay, so by way of introduction, I'm Andy Spakowitz. I'm a professor in chemical engineering and material science and engineering at Stanford. I'm also the senior associate dean of engineering at Stanford, and so it's a real pleasure to be able to give this talk. So thank you so much. Oh, there we go. So, at the heart of a lot of the work that we do is we aim to develop a fundamental understanding of epigenetic regulation. So I am a polymer physicist by training, and DNA is a very long polymer, and the way that DNA is organized and structured in living cells is very important with regard to how the genes are expressed, how the proteins are expressed in a timely manner, and how that is controlled. Now, the mechanism by which gene expression is controlled in eukaryotic cells and complex multicellular organisms is called epigenetic regulation. And the basic premise is that you have the same genes in all of your cells, but they have very different behavior in different cell types. And the reason for this is that it's all has to do with how the proteins are expressed. Now, from my perspective, if you think about, well, how do you control how the proteins are expressed, there has to be some sort of, additional programming. And that programming essentially, from a very simple perspective, is driven by those genes that you want to silence. You sequester them, you condense them into regions, and in this case, they're called heterochromatin. And then those that you want to express are in more open and accessible regions called euchromatin. And if you look at a cell stained for DNA that on the right, you can see these dark regions. Those coincide with those genes that are silenced, and the light regions are the ones that are expressed. So in many regards, it's a very physical phenomenon, right? You have to kind of physically sequester these genes in specific regions within the nucleus. And the way that this is done is through these chemical marks that are made either to the DNA itself or to the proteins that package the DNA. And our goal is to develop some physical insight into how that packaging takes place and what are the consequences on gene regulation. So, but what I'm going to do is I'm actually going to start with a very historical measurement. Okay, so I already alluded to the fact that the way that proteins engage the DNA is very important. And so let me tell you about an amazing set of measurements that were done now, over 40 years ago, by Berg, Winter, and von Hippel, which were basically to try to understand how a protein is able to find a specific site on DNA. So in this case, they measured lac repressor on DNA, and these were in vitro experiments. And what they found is that the measured rate of binding, okay, so the apparent rate of binding of lac repressor to a specific site is on the order of 10 to the 10th per mole per second. Okay. Now that number, that's just a number, but to put it into perspective, is if you considered it to be purely a diffusion controlled reaction in 3 dimensions, then you would anticipate that the effective rate of binding would be about 10 to the 7th to 10 to the 8th per mole per second. Now that is a huge discrepancy, right? So there's something crazy happening. There has to be something that gives rise to this large discrepancy. And furthermore, if you look at the plot on the right, by the way, can you see my cursor as I'm moving it around? Okay, great.
Yeah.
If you look at the plot on the right, this is a measurement of the apparent rate of binding of lac repressor versus, in this case, it's salt concentration, but that's kind of a stand-in for the affinity of the lac repressor to the DNA. And you see that it's non-monotonic. Okay, so there's actually some optimal binding affinity that gives you this apparent rate, which is quite high. And to be clear, this is log of the apparent rate. So this is actually orders of magnitude, variation that we're talking about here. And so, okay, what was proposed by Berg, Winter, and von Hippel at the time was 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 really quite fascinating. The idea that perhaps what it amounts to is that the DNA is not just diffusing around and then combining to the binding site right when it happens to be in the same, spatial proximity, but that it uses non-specific binding to the DNA, to the rest of the DNA, in order to alter the path that it takes to get to the binding site. And ultimately, it's a more efficient way for it to find the binding site. And in fact, it's kind of akin to an antenna. The idea that there is some physical mechanism that by non-specifically binding, you will translocate and that there will be ultimately an effect where it reaches that target site faster than what it would if it was just going in 3 dimensions. So that was proposed back 40 years ago, which was incredibly intuitive on their part. And so the way to look at it is that they identified four main mechanisms for how it would translocate along DNA. There would be sliding, where it's non-specifically bound and it's sort of sliding around on the DNA. There would be these micro hops where it pops off the DNA. It diffuses a little bit, but then it rebinds in close proximity. There are macro hops where essentially it pops off the DNA. And then because the DNA is this sort of flexible, wiggly object, that it would diffuse a short distance, but it could go a very large distance in genomic position. And that's a macro hop. And then they also identified intersegmental transfers, where if there was a loop in DNA, it could just sort of slide and then just transfer to a distal site without, too much, effort. Now, why is this important? So a couple of things. What it points to is that if you have structure in the DNA, that will alter how the proteins translocate along the DNA. So that's an important point. What it also does is it gives you this sense that, that the physical mechanism for how the protein engages the DNA will alter the translocation and the search process. Okay. So essentially what we endeavored to do at the time was we wanted to develop a theory where we can incorporate the details of the chromosomal organization into the theory to understand how proteins can find a target site. And in that case, back in 2010, when we first started publishing in this space, we were really thinking about regulatory proteins like lac repressor. In the second part of my talk, I'll explain why it's related to epigenetic regulation. But for now, we're just focused on lac repressor. Okay, so we what I'm going to talk through is the foundational principles for a transport theory that can predict the large-scale translocation of proteins along DNA based on the statistical properties of the DNA, the structure of the DNA, as well as the kinetics for binding and unbinding and translocation of the protein itself. Okay. And to be clear, there's some foundational math and physics that I have in this, and I'm gonna kind of walk you through the basic elements of it. A lot of the details I'm leaving out, but I want to make sure that you have a sense of how one develops an analytical theory that can address genomic scale DNA at timescales that are relevant to experimental measurements. Okay, and that's a challenge given that DNA is just so massively large, and the timescales that we're talking about are quite large as well. So here's the basic premise. Now, if you think about if you strip down all of the picture that Berg, Winter, and von Hippel had put together, what you need is you need to have a transport theory that has sort of different states for how the protein is moving at a given time. And simply put, we're gonna simplify the whole picture and just simply say that there is a statistics for how proteins move along DNA while it's bound to the DNA. And in that case, we're going to be very simple. We're just going to say that's a one-dimensional diffusive process along the DNA. And then there's a transition kinetics that leads to the DNA or the protein popping off the DNA. And then what happens is once it pops off the DNA at this site right here, the protein diffuses in 3 dimensions, so it's moving around in 3 dimensions, and it will re-engage with the DNA based on what is the statistics of the DNA configuration. Okay, so while it's off, it's got a combination both of 3-dimensional diffusion of the protein as well as the conformational properties of the DNA that it popped off of. Okay, so let's break this down just a little bit. So mathematically, if one is to describe the diffusive transport along DNA while it's bound to the DNA, it's a one-dimensional diffusive phenomenon. And that's very well described, that has a long history, and and Smoluchowski, wrote the equation that would govern the diffusion along the DNA. And essentially, this will obey a Gaussian random walk along the DNA. And that's governed by an on diffusion. And of course, the time that you spend while bound to the DNA is going to affect how broadly it explores the DNA while it's sliding along. Okay. So the off case is really a little bit more complicated. Okay. So let me walk you through. What I already alluded to is that when the protein pops off the DNA, the protein itself diffuses in 3 dimensions. And so that's governed by a statistical probability, which is diffusive as well, but now it's in 3-dimensional space. Okay. Now, the DNA, it is governed by some chain statistics. Okay, this is what you would derive from polymer physics, and there are many models for describing, different polymers, and I'll talk about how one would capture a in vivo configuration later in this talk. But for now, let's just say that it obeys something a polymer model called the worm-like chain model, which, for the record, is very suitable for describing DNA, bare DNA, naked DNA in vitro. Okay, and that is the basis on which we're comparing to the experiments. So we're just going to use a simple worm-like chain model for describing the statistics. Now, essentially, for you to describe the probability distribution while you're in the off state, you need to have both the protein diffuse a certain distance to a position R2, and the DNA itself has to have a segment which is located at another position R1. And if R1 and R2 are within some distance A, then they can recombine together at that point. And so the statistics of this is governed by a combination of both the protein and the DNA. Okay. Now, the last little bit to pull the whole story together is that you need to have statistics for the kinetics of binding and unbinding. It has to transition. Now, the simplest way to describe the statistics for unbinding while you're on the DNA and you want to find what is the probability that you unbind at some time. The simplest way to describe this is just to say that it's governed by just a first order reaction rate, okay, with an unbinding rate constant of Ku. Okay, so this is not surprising. This is sort of what you would expect if there was a characteristic rate of unbinding, which is independent of time. Now, the kinetics for going from the off state to the on state, or in other words, rebinding, is a little bit more subtle. And the way to describe it is to just simply say that the probability that you remain in the off state as a function of time is governed by a binding rate constant. And then this M is the total length of DNA that you encounter as a function of time after you unbind. Okay, but essentially this is just a well-described statistical distribution that one can describe with the DNA configuration and the protein diffusion. Okay, so all the math is done. That's all that I wanted to say about the foundational math, but I wanted to give you a clear enough picture of what it was that goes into this. But suffice it to say that essentially what we have is we have this combined transport model that combines on state and off state and the kinetics in between. And then, oh, I said we're done with math. I'm going to just do a little bit more math. One little other thing, just in order to close the whole thing up, is that in order to describe the phenomena, which is called facilitated diffusion, that is the model that was laid out by Berg and von Hippel, one has to then say, if I want to translocate from this site here at the beginning to this site here, you have to have the combination of every single trajectory that you possibly could imagine. And that would include sliding, popping off, rebinding, and then sliding. That would be a three-step phenomenon. A four-step phenomenon where you are on and then you're off and then you're on and then you're off. And you have to enumerate every single one of these possibilities in order to get the full distribution for the probability. Well, thankfully, that mathematically can be described. And with a little bit of work, you can say, well, the total probability density must be the combination of just being on plus going on and then off and then being on and off and on. And you just enumerate all of those. And the beauty of that is mathematically, it just reduces to a simple form, and boom, you've got a statistical distribution which governs the position and time for the protein along the DNA. Okay. And when you work out all the math, it has a very nice, clean form. And this is the governing equation for facilitated diffusion that we found based on these on-off events, and it allows you to describe how the protein would move along the DNA. And so the question is what does it look like? Okay, so let's take a look at a couple of things. The first thing to note is that if you look at the distribution while it's in the off state, so this is the probability that the protein, if it leaves the DNA at time 0, that at 5 different times later, it will re-enter the DNA as a function of position. So at very, very short times, and this is perhaps the most intuitive one, this blue curve here, you see that there's a high probability that you re-enter the DNA very close to where you left. Now, the reason for that is because it's like a micro hop. If you hop off the DNA and you diffuse just a little distance, you're probably going to re-enter right at that position. Okay, but then you notice that the probability drops down orders of magnitude, but then it has this weird little peak up here, and then it goes down again. So what's this peak? Well, it has to do with the fact that if the DNA happened to have if in the DNA statistically it had a loop, then it can re-enter at that location where the loop happened to be. And so that is what would be described by Berg and von Hippel as a macro hop. Okay, so then the probability goes down dramatically because the probability of a loop at much larger distances gets negligibly small, and that has to do with the statistics of the DNA and the fact that it's a random walk at very large distances, so it goes away. Okay, and as time progresses, as you go to longer and longer times, the cloud of the protein statistics diffuses out, and eventually it just sort of, will sort of evenly go back onto the DNA at larger distances. Okay. So what you can do is you can then say, well, how fast does the DNA effectively translocate along, or how fast does the protein translocate along the DNA? And the way that we would describe this is by calculating the mean displacement. Okay. Now there's some subtleties that I don't want to go into mathematically. It turns out that because of the statistics of the DNA, the protein tends to transport super diffusively because it obeys something called a Levy random walk. I'm not going to talk about the math right now. It's quite fascinating. It has a long history. It's very cool, but I'm not going to talk about it. Okay. But nonetheless, what you find is that notice if you plot, this is a surface plot of the super diffusion coefficient, or simply put, how fast the protein effectively translocates along the DNA. And what you find is that if for a fixed unbinding rate, and you alter the binding rate, or in other words, the binding affinity to the DNA, notice that the superdiffusion coefficient goes up as you go along. There's an optimal value, and then it goes back down. So in other words, if you were to change the binding affinity, there's some optimal binding affinity where it peaks and then it goes back down, which is very much in line with the observations by Berg and von Hippel. Okay, so that's cool. Now there's another effect, which is important, which is to say, what is the ratio of the length of distance that you're on the DNA and the while you're off the DNA? Because if you think about it, if the distance that you're on the DNA, the footprint that you're on, is much smaller than the distance that you're off, then you're skipping over a lot of the DNA and you're going to miss a bunch of stuff. You're not actually exploring it all, right? But of course, if the distance while you're on the DNA is much larger than off, then you're revisiting the sites many, many times. So there's some optimum. And that ratio, which would be optimal, would be a ratio of 1. And that is this dashed curve right here as a function of unbinding rate and binding rate. So interestingly, if you look at a bunch of proteins, and in this case, they're mainly repressors, but well, lac repressor, but also restriction enzymes, and other repressors as well. You find that they tend to be sort of in this sweet spot between being optimized for speed and being optimized for efficiency. Okay. And then, so that's very anecdotal. But if we take our model and then we use some sensible numbers for the binding affinity, okay, you indeed can reproduce the Berg and von Hippel observation for lac repressor, where at low, whoops, sorry, at low salt concentration, okay, or high binding affinity, you wind up having this plateau, and then there's an optimum, and then it falls off dramatically when the binding affinity goes down. And if you look at trajectories of these little dots are the protein, okay? If you have a very high binding affinity, okay, so that's this first one, it just slides along the DNA, so it slows the process of finding the target site because it's always bound. When you get to this optimum, 0.2, you see that it's sort of balancing the on and the off. And then eventually, when you go to very high salt or low affinity, then it sort of is just diffusing in 3 dimensions. It never actually finds the site. Okay. So that's what I wanted to say about the sort of fundamentals of facilitated diffusion in the context of lac repressor. And again, this was originally developed. This was theory that we developed now, almost 20 years ago, mainly to understand that regulatory protein. Okay, so I'm going to pause here. That's kind of the first part of my talk. And should I take questions at this time, or how is this normally
So
done?
that would be great. Yeah, if people have questions. I actually had one clarification question. When you say that the motion of the protein is super diffusive, you mean super diffusive in 1D, right? Or is
Yes.
it? Yeah.
Yeah. Yeah. It's super diffusive in 1D. Yeah. So that's very important. The basic notion is that because you have these really long tails in the hop distance distribution, and
Mhm.
because they're power law tails, that leads to these really fast, super diffusive motion, but on the one dimension of the chain. Yes.
Mhm. No, that's fascinating. Yeah, no, thanks for a beautiful exposition of this background. Gregor has a question. Gregor, go ahead,
Yeah,
unmute
hi.
and
Thank you for the presentation. So the question I have is, um, how do you apply this to DNA where you actually have chromatin on there? And one question. The second question is, in your diagram, you showed like one strand of DNA, but in eukaryotic cells, in most cases, except the haploid yeast genome, you have two copies of the genome. And so I wonder,
Yeah.
how would the theory apply there, and could you make modifications?
Yeah, absolutely. This is a great question. So I'm going to talk about in the second part of my talk, I'm going to talk about DNA methylation, okay, where there's a methyltransferase that is essentially translocating along chromosomal DNA in living cells. And the way that we approach it, I'll describe, is that, we extract from what is measured experimentally the statistical distribution of the DNA based on the high C data. There's essentially a genome averaged distribution for probability of the DNA based on the in vivo measurements. Um, what I will say is that a lot of what we're talking about is right now our approach is very averaged, but and today I'm not going to talk about other simulations and theory that we've developed, which actually incorporate the heterogeneity associated with euchromatin and heterochromatin. That's another area of research that we do. Down the road, we anticipate that what you really want to do to capture chromosomal statistics is have a more detailed model that captures that heterogeneity. So I'm going to be very genome averaged when I talk about the behavior in vivo. But I would say that down the road, building in more precision that captures the heterochromatin, euchromatin, and the heterogeneity is absolutely essential. So that was the first question. Sorry, what was the second question? I apologize.
Sorry, the second question was, so this model is like for one strand of DNA, but like what happened in reality you have two strands. And in general, like high C data doesn't really, at least to my understanding, doesn't really distinguish between that. So
Yeah.
there's always the assumption that this has all happened on one strand, and I was wondering how valid this assumption.
Yeah, that's true. That's a good point. So first of all, since the particular model that we were developing for lac repressor, this was to capture the in vitro measurements. It's really, it's very simplified relative to that. I do think that, capturing in vivo effects, including, the two copies of the genome, um, and territories and other things, that there's a lot of steps that have to be taken. And in this talk, I'm not gonna take those steps because we're really trying to get those very averaged, picture. But I think that given how the community has developed such great, platforms for describing genomic DNA and some aspects of the territories and how, the copies actually arrange themselves. I'm hopeful that that can be built into the model, but that's down the road.
Thank you.
Yes.
great. So maybe we should continue with the second part of the talk, Andy.
Sure. Yeah,
Yeah,
absolutely.
great.
So, actually, so Gregor, your questions, let me give you let's now talk a little bit more about the in vivo chromosome, which is, if we want to make progress and ask ourselves, well, how do proteins that engage the DNA and either condense the DNA or write the epigenetic code, how is that related to the multi-scale nature of the chromosome? I mean, we've already, in the very simple example that I talked about in the first part of this talk, I relayed that the multi-scale nature of DNA, even just in vitro, is very important for dictating how a protein would translocate along the DNA. So, in my lab, we're very interested broadly in understanding the full picture of chromosomal DNA and epigenetic regulation in living cells. And we've sort of identified various problems at different length scales of relevance. So, for example, on a very local length scale, the question of how the nucleosomes and the chromosomes are arranged locally, how they affect epigenetic modifications, how those modifications are written, how they are recapitulated over multiple cell generations, these sort of questions. It really has to do with the interplay between reader proteins and writer proteins, and how they engage the DNA as a function of the marks that are along the chromosome. Now, at a intermediate length scale, we're also quite interested in questions of how chromosomes reorganize. Okay, so looping, the loops of the DNA, we've already, identified as being important for understanding translocation and how proteins move along the DNA. And what we want to be able to do is understand how those loops are made, how they move along and reorganize as a function of time, and how that's related to the formation of complexes, for example, regulatory complexes, because enhancer promoter interactions are essential for driving gene expression. Now, there's another effect that I will point out. I'm not going to talk about it today, but from a range of experiments, what is found is that actually a lot of these processes for reorganization are not just based on thermal fluctuations alone, but there's the additional effect of how proteins drive translocation of the DNA. And in fact, if you modulate ATP in the cell, it will dramatically influence how those chromosomes move. And so we want to understand what are the foundational principles that dictate how chromosomes move around dynamically. And then at a much larger length scale, where you get these either, gene territories or condensed regions in heterochromatin versus euchromatin, we want to understand the foundational principles that drive that condensation and how that affects the accessibility of the DNA as a function of the distribution of marks along the DNA and the proteins that package the DNA. So for today, I'm actually going to talk more about epigenetic modifications. And in fact, I'm going to focus my discussion almost entirely on DNA methylation. And then I'll comment very briefly on histone modifications at the end. But primarily, I'm going to focus on DNA methylation. Okay. So let's talk a little bit about DNA methylation. So the organization, transcription of DNA within a cell is determined by epigenetic marks. Okay, and one of those marks, which is very important, is the methylation of DNA itself. Okay, this is a very foundational mark. It is extremely important in gene regulation. It's also very clear that when you get aberrant methylation patterns, that it is linked to diseases such as cancer and genetic disorders. Okay, so that so obviously there's a genetic component for these diseases, but the epigenetic component, if you have alterations in patterns, this is a prevailing effect that arises in these diseases. Now, there are, of course, in order to lay down the marks, there has to be certain proteins that add the methyl mark to the DNA, and this methyltransferase adds a methyl group to what are called CpG sites, or in other words, cytosine guanine dinucleotides. Okay, now there's there's, the patterns of methyl marks in DNA show dramatic changes from, different cell types and more specifically to diseased and healthy cells. So here, I'm just showing you a picture, and this is or a plot of what is genome averaged correlation in the DNA methylation as a function of various cell types. And these happen to be a range of different cancer cells. And what you see is that, and I'm going to go into the details in a little bit, but just suffice it to say that there's characteristic, genome average differences in how correlated the DNA methylation is in these various cell types. Okay. And what I'm going to do in this part of the talk is I'm going to walk you through how the transport of the DNA methyltransferase, okay, how that can be modeled using a facilitated diffusion model, which we had originally developed for understanding lac repressor binding, and how that relates to the varying patterns that you see in these cells. Okay, so the DNA methyltransferase, DNMT, and there's various DNMT numbers, okay? But I'm just going to broadly, since we're just talking theory, I'm going to make it very simple. This is just a DNMT. We know that, DNMT has some non-specific binding to the DNA itself, okay? And so, as you can kind of imagine, if you are DNMT and you are, let's say that you happen to be engaged on the DNA at some location, that if you have a non-specific binding to the DNA, the way that you could statistically lay down methyl marks onto the DNA itself is you can diffuse along the DNA and undergo this sliding transport. And if you encounter a CPG site, you have some kinetic for laying down a methyl mark. And then eventually, because it's non-specific binding, you may pop off the DNA and diffuse in 3 dimensions, and then re-encounter the DNA at a distal site, and then lay some down. So the correlation of these methyl marks would be influenced by the transport behavior of the methyltransferase along the DNA. So the basic principles that we laid down for understanding the lac repressor may be relevant to understanding the mark distributions that you see in these cells. Okay, so here, what I'm going to do, and so there was this great question earlier about, well, DNA in living cells is more complicated, right? And naturally, having a model that captures this sort of multi-scale behavior of DNA is really important. Now, because we're doing genomic averaged behavior in this initial study, I'm going to be very simple. We're just going to take our theory that we developed for lac repressor, and rather than using a classical polymer physics model, which may be relevant for in vitro measurements, we are instead going to just simply take what is extracted from an average of Hi-C data for the statistics of the polymer chain. And very, very simply, what you see is that if you look at the Hi-C data, and if you were to average as a function of distance away from the diagonal, what you see is that there is a short distance behavior, which appears to be, somewhat random walk-like and folded at a local scale. But then there's this intermediate scale in genomic distance where the effect of cohesin will cause there to be these sort of loops. And what it essentially says is that if you average it out, that there's almost a plateau or a more shallow scaling that you see for the mean displacement of the DNA versus genomic length. But then eventually at much larger length scales, it goes back to its sort of walking around. Now, this is the most simple possible way of describing the chain statistics based on a a more detailed model extracted from experiments. And this is consistent with that sort of averaged behavior. Now, suffice it to say that, of course, capturing the actual genomic organization would be a much more sophisticated way for describing methylation patterns as a function of genomic position. But for now, we're just going to suffice it to say, since we're modeling genome averaged behavior, we're just going to have a genome averaged statistical description of the DNA, and further refinements will be taken down the road. Okay, so here's the thing, is that if you take a facilitated our model for how the proteins how this methyltransferase would undergo a local diffusion along the DNA, it would periodically hop off and translocate in 3 dimensions and undergo this sort of transitional statistics between on and off states. What one can do is you can do a calculation where you calculate what is the total exploration time at each site along the DNA if you started at position 0. Okay, so in other words, let's say that at position 0, you laid down a methyl mark. Okay, so you're on the DNA right at position 0. And then you ask, now the protein is going to diffuse away. And you ask yourself, well, what is the total time that you spend at each site as time progresses? So in other words, another way to think about this is a plot of the fraction of the time that you spent on each site as each time goes forward. So if you're at a very short time, right, you're going to spend most of your time in close proximity to where you started, right? And then maybe there's like this tail, and you notice that this tail has these different scaling regimes. Well, that has everything to do with the statistics of the polymer chain that we fed into the model, that it has a local scaling associated with the local organization of the DNA. This intermediate regime has to do with that intermediate scaling associated with cohesin. And then at large length scales, it goes back to this other scaling. And as time progresses, you actually spread the protein will spend more and more time at positions that are further away from where it started, and it will start to plateau. So of course, as time continues, it will sort of even out how much time it spends at each site genomically at distal sites. Okay. So if we then just simply assume that when it encounters a position, that the probability that it will methylate that position is a function of both the time it spent at that position and a methyl transfer rate, okay, just a simple rate constant for methyl transfer for the methyltransferase, then you would get that the fraction of methylation exhibits this statistical behavior, that it has this plateau, and then eventually it goes down, and it's going to go down based on the statistics of the polymer chain, and it has these various regimes. Now, what I'm already going to point out is that if you remember that data for the methyl correlation, it had these really long tail in the methylation. And in fact, it kind of went down, and then it had a flat behavior, and then it had this long tail. We're already beginning to see a remnant of that behavior, and I'm going to show you how this all works out. But there's one other thing that we have to point out, okay? Right now, we're assuming that every single site on the DNA could be methylated, but that's not true. It only methylates CPG sites, okay? So we got to incorporate a little bit of genetic information, okay? So if I'm going to say the probability of methylation, P sub m, is going to be a combination of two things. One is the probability that a site is actually a CPG site, okay, so that has to be true, times the fraction of the CPG sites that would be methylated, or the fraction of methylation that would occur by DNMT. So the combination of those two has to conspire together to give you the total probability of methylation. Okay. So it turns out that genetically, there are these things called CPG islands. So generally speaking, there are regions that tend to be about 1,000 base pairs long, and those regions tend to have at least 50% of CPG density. Okay, so there are these sort of hotspots for CPGs. And the simplest possible description of how those sites would be correlated is going to be governed by an exponential distribution, which has a scale which is on the order of 1,000. So the average size of a CPG island based on this correlation would give you this region that's about 1,000 base pairs. Okay. So the total methylation correlation, if we want to make a direct comparison to experiments, is going to be a combination both of the CPG site correlation as well as the methylation correlation associated with the facilitated diffusion of the methyltransferase. Okay. So the total equation for the methyl correlation, it combines both the correlation for CPG, C sub C, and the correlation associated with the methyltransferase, which we call C sub F. Okay. So this model now, and here, in a talk like this, I tried to walk through all the fundamentals of it, but I think it's important to take stock in what is the theoretical prediction dependent on. Okay, so it turns out that the theoretical prediction is actually dependent on four parameters only. Okay, it's the DNMT transport, and in this case, you can reduce the DNMT transport just down to the ratio of the length while it's on versus the length while it's off. That's the only quantity that it depends on, the ratio of those two things. There's a parameter associated with the DNMT binding kinetics. There's a CPG distribution, but this actually is a fixed quantity because we know that the distribution is just governed by the CPG islands being 1,000 base pairs. So this actually is not a parameter, but it's a fixed quantity that we just set based on what's observed genetically. And then there's a methylation fraction. Okay, so that's a total fraction of the sites that are methylated. And this can also be parsed in terms of a methylation rate. Okay, so really there's three parameters. And so the data here is what I showed earlier. This is from the paper by Zhang et al. And what you see is that when you look at the methylation correlation for each of these cell types, and let's just focus on this one right here, is that you notice there is a fast fall off in the correlation, but notice that happens over about 1,000 base pairs. So the source of that is simply just the CPG correlation. So if the only thing that mattered for methylation correlation was the correlation in CPG sites, you would get a plot that would go down like this, but then it would just keep going down. Okay, so these long tails has something to do with how the methyl transfer is correlated at very large distances. And we assert that these tails right here are entirely associated with the facilitated diffusion model for the methyltransferase. Okay, so if you take our model and you take the parameters, and the parameters actually don't vary dramatically, but if you vary those parameters associated with the transport, the methylation kinetics, and the fraction of the methylation. Okay, so these guys are affected by each other, but nonetheless, these are three distinct parameters that go to the model. You see that you capture these elements of the features of the experimental measurements, specifically all of these have this short fall off associated largely with the CPG correlation. And all of these tails, the different regimes, so you can actually sort of see remnants of this intermediate regime and then the long regime, that sort of picks up on the chromosomal organization and how the methyltransferase translocates along the DNA as predicted by our model. And so the basic gist of what we're trying to achieve here is to just simply lay down the fact that the organization of the DNA plays this essential role in dictating how the short range and the long range methyl patterns arise in these cells. And the variation from different cell types is indicative of variations in how the performance of the methyltransferase is going to influence the way that those sites are laid down. Now, moving forward, of course, we want to be much more sophisticated and say, well, how would this specific CPG site get altered as a function of the organization of the DNA? And that is going to require us to think about the interplay between how the heterochromatin and euchromatin are arranged and how that is related to the transport behavior of the methyltransferase. And that's something that we're thinking about actively now. So what I'm going to do now, I had a couple of slides that, depending on the time, I was considering talking a little bit about how histone modifications are laid out and how that is related to the organization of the DNA. But let me just very briefly just say that, the histone modifications has a very interesting interplay between the reader proteins. In this case, we're talking about HP1, which binds to a particular methyl mark on a tail of the H3 core histone protein, and the methyltransferase associated with that methyl mark. But basically, it has to do with how the binding of the reader interacts with the writer protein and how those two play into one another. It's a very sophisticated, really interesting set of problems. But what I'm going to do now is, instead, in the interest of time, I'm just going to simply say that that aspects of the chromosomal organization and the binding of those reader proteins, the interplay between them, can capture experimental measurements of how those sites are methylated as a function of distance from a point where the reader protein happens to be bound. So instead, what I'm going to do is I'm going to pause, because I want to make sure that we have time for discussion or questions. And let me just conclude by saying that, in the work that I described today, I started off with some historical data by Berg, Winter, and von Hippel, talking about regulatory proteins and how they utilize transient DNA binding to accelerate genome exploration. And that there's an optimal strategy for target site localization, which balances periods of being bound to the DNA and unbound to the DNA, which explains this anomalous behavior in lac repressor. And then what I did today is I made the argument that these mechanisms for how proteins translocate on DNA can be used to interpret how the marks, epigenetic marks, are actually correlated in living cells, and in particular, variations in those correlations that arise in cancer and in healthy cells. And so basically, the picture is that as we think about chromosomal organization and how proteins engage those chromosomes, that's very important for understanding how marks are established and how the genes are organized and ultimately expressed. So of course, I want to thank there's many students that contributed to this, but the main students that I want to point out is that in the first part of the talk, Lena Koslover and Mario Diaz de la Rosa were the primary drivers for developing the theory for lac repressor, and they did really beautiful work This was earlier in my faculty career. And more recently, as we revisited and tried to understand epigenetic regulation and the marks, a really great student, Ariana T., sort of rekindled that work that we did on facilitated diffusion and used that as the basis for understanding the DNA methylation patterns that I described in the second part of my talk. I received funding from National Science Foundation, both from the Physics of Living Systems program and the using the Rules of Life program for genetic mechanisms. So thank you very much. Oh, I should also say that some of the collaborators that were really important on this, I should point out that in the using the Rules of Life work, John Allard, Tim Downing, Elizabeth Reed, were really great to work with. And thinking about DNA methylation, I'm really grateful for their collaboration. And that concludes what I wanted to say, and I'm happy to answer any questions.
Great. Thank you so much, Andy. That was a absolutely fascinating hour. I was totally blown away by how you have a polymer physics model and you fit that data so well. But anyway, we'll come to that in the questions. First, let me clap on behalf of the audience. Folks, please type your questions in the chat and or raise your hand, and I'll just call on you to unmute and ask the question yourself. I had before we start with the questions in the chat, I just had one question again about the data you present, the predictions of the model. Just a couple of slides back compared to the correlation of epigenetic marks. Is that a fitting, or is that something which is more like, you estimated parameters and you plugged it into the equations?
Yeah, I mean, it is a fitting. There's definitely, like, you do see the variation, for example, in the fraction methylation, and you see, quite significant changes in fraction. So it is okay. So these are fitted. The three parameters are fitted on the individual data sets. They appear to be pretty sensible. So for example, the methylation rate, it varies a little bit, but it's sort of within a certain range. I mean, you can look at that. You do see, so ostensibly, we did try to do a fit where we just stuck this as some number, just did a global value, and then did the other ones. And you get reasonable fits, but not, there's some variation. And there's some argument for why there would be some variation in the cell types. But it is observed that the overall global fraction of methylation does vary from cell type to cell type. And that actually is a big driver for these variability. So I think that's we didn't go into trying to interpret the differences too much because I think we want to try to get to a more granular picture of specific site methylation as we move forward.
Cool. That's yeah, fascinating. Guangxi, you had a question. Do you want to unmute and ask it? Or should I ask it on your behalf? Yeah, go ahead.
Yeah. Can you hear me?
Yes.
Yeah. Oh, excellent talk. So I'm wondering, so I assume that in all the theoretical calculation for the target search of protein, you're assuming the kind of annealing case for the chromatin dynamic itself. So you're considering like on the average sense. So I'm wondering, have you considered how does the dynamics of chromatin itself play a role in the target searching of the protein?
Yeah, that's a great question. So you're absolutely right. This is based on a statistical argument for how the DNA is arranged. And there would be an effect where the non-equilibrium dynamics of the actual dynamics of the DNA simultaneous to the dynamics of the protein would affect things dramatically. So we looked at this in the earlier work that we did on lac repressor. Okay, so we did like simulations of the DNA in at the same time as the protein is translocating. We didn't see a dramatic influence. I think that there's a long okay, so there's a bit of history on this. If you think about looping, DNA looping statistics, it turns out that like if you go to very large genomic distances, the looping kinetics are governed by non-equilibrium concerns, which are more genomically related to something called the Rouse time, which is the longest relaxation time of that polymer as a function of distance. So if you go to really long genomic distances, then yes, I would anticipate that that should emerge as a bigger effect. The in vitro work that we did was based on 50 kilobases because this was lambda phage. And so it didn't emerge as a major player. But it's a good question. I mean, I think it's something that we haven't thought about in vivo. But you know, there's of course lots of questions, and that's certainly one of them. Yeah,
Okay, thank you very much.
absolutely.
Matthias, do you want to unmute and ask your question?
Yeah, I'm going to give it a try. So hope you can hear me. So great talk, Andy, as always. So concerning the plot that we see right on the screen, so I guess transport only becomes important if you have few enzymes that do the work, because otherwise there's always an enzyme next to the point where you want to do some work. And so transport, so you're in a well-mixed system then basically. So can you therefore back conclude by fitting to your data what is the effective amount of real enzymes that need to do the work? So how limited the system is in terms of workers?
I mean, I think it's a great question. I think that, you know, there's a couple of things that I can say. Uh, we no, I mean, let me first say is I think it's a very good suggestion. I think that actually having taking more stock in the occupancy and what's more you know, consistent with the complexity in vivo is something that is, you know, is of course challenging. I mean, I realize that we are very, you know, we're kind of we're of course reductionist, right? Like I want to have the simplest possible picture and just draw a conclusion from it. But I think your point is very well taken, and I think that having better stock in the local environment and the proximity of other proteins, and using this as the basis for making some arguments about what is needed and what isn't, in order for these things to work, I think would be a really good direction to go. Yeah. Thanks.